Calcium uptake in plants is a fundamental process that occurs primarily through the root system. Understanding how plants absorb calcium from soil and the factors that influence this process is crucial for plant nutrition and agriculture. Calcium uptake begins in the soil solution surrounding plant roots. The root system, including fine root hairs, creates an extensive surface area for nutrient absorption from the surrounding soil environment. Calcium uptake is largely a passive process, meaning it doesn't require the plant to expend energy directly. Instead, it's driven by the transpiration stream, the movement of water from roots to leaves as water evaporates from leaf surfaces. Calcium enters root cells through non-selective cation channels in the cell membrane. These channels allow positively charged ions, including calcium, to pass through the membrane and enter the plant's vascular system. Several key factors significantly influence calcium availability and uptake in plants. The concentration of calcium in the soil solution directly affects how much calcium is available for absorption. Soil pH plays a crucial role because it affects the chemical form and availability of calcium ions in the soil solution. Acidic soils can reduce calcium availability. The presence of other cations like potassium and magnesium can compete with calcium for the same uptake sites, potentially reducing calcium absorption when these competing ions are present in high concentrations. Soil aeration and water availability affect root health and transpiration rates, which indirectly impact calcium uptake. Well-aerated soils with adequate moisture support better root function and calcium absorption. Mycorrhizal associations with beneficial fungi can significantly enhance calcium uptake by extending the effective reach of the root system. These fungal partners help plants access nutrients from a much larger soil volume than roots alone could reach. Understanding these mechanisms and factors is essential for optimizing plant nutrition and addressing calcium deficiency issues in agricultural and horticultural systems. Once calcium enters the plant through the roots, it begins its journey through the plant's vascular system. The primary pathway for calcium transport is through the xylem vessels. Calcium moves upward through the xylem along with water and other nutrients, reaching various plant tissues including leaves, stems, and developing fruits. However, calcium has a critical limitation in plants. Unlike other nutrients, calcium is relatively immobile in the phloem, which means it cannot be easily redistributed from older tissues to newer, rapidly growing parts. This immobility leads to calcium deficiency symptoms appearing first in rapidly growing plant parts, such as young leaves and developing fruits, even when older tissues have adequate calcium levels. Calcium plays several crucial roles at the cellular level. First, it is essential for cell wall structure, where it forms calcium pectate compounds that provide strength and stability to plant cell walls. Calcium also maintains membrane stability by regulating membrane permeability and preventing cellular damage from stress conditions. Additionally, calcium serves as a crucial secondary messenger in enzyme regulation and cellular signaling pathways, helping plants respond to environmental stresses and coordinate growth processes. Adequate calcium levels are essential for fruit quality. Calcium deficiency can cause serious disorders such as blossom end rot in tomatoes and bitter pit in apples, which significantly reduce crop value. Calcium accumulation in plant tissues is influenced by several factors, including transpiration rates in different plant parts and the plant's overall calcium status. Higher transpiration rates generally lead to greater calcium accumulation in those tissues. Understanding calcium accumulation and function in plant tissues is crucial for managing plant nutrition and preventing calcium-related disorders that can significantly impact crop quality and yield.